Okay. I've looked at it on Google Earth. It's pretty interesting.
Okay.
Here we go.
All right, we're gonna get started. All right, hello everybody again. I think I met a lot of you last night. If not, I'm Ellen Currano, University of Wyoming, botany and geology. So I have the split scientific personality. I do a lot of work in Wyoming and then also a lot of work in East Africa. So we did the Wyoming thing last night, and today it's just kind of going to be start off with a travelogue because Ethiopia is just an amazing country, and so to show off some of the wonderful experiences there, and then a little bit about the science that we do. So this word came up last night, the Lagerstätte, so the German word just meaning incredible fossil deposit, and you'll see why this one is so much better than what we talked about last night. Okay, so we have the African continent. Let's try this thing. Okay, and so Ethiopia would be kind of in that general area right there. So it's a landlocked country. You have Eritrea to the north, which is along the Red Sea, and then Somalia over here, and Kenya to the south. Let's see, I started working in Ethiopia in 2008. That was January of 2009 was the first time that I went. And I was, for like 7 years in my life, I would go over there for a couple months a year. I also went on sabbatical there and got to spend 5 months living in Addis Ababa, working at the museum and seeing the country. And it really is an amazing country. So kind of, starting off with just some classic scenes of Ethiopian country life. So this is one of my favorites. You unfortunately see all over the countryside women doing the heavy labor, collecting. A lot of times it's wood for charcoal. I'm not sure what she's carrying. It's probably not burning wood, but some sort of plant material to be used or to be sold. And then so Ethiopia is the source of the Blue Nile. Lake Tana is there. So beautiful scenery around there. Okay, so we know, probably most people know Ethiopia, the rift cuts through it, but you also have these incredible highlands. So this is northern Ethiopia. It's the Simien Mountains National Park. I think Ethiopia has more UNESCO World Heritage Sites than any other country. This is one of them. And so about 30 million years ago, 2 kilometers of basalt were emplaced there. So you just have, tons and tons of basalt, these beautiful cliffs. You'd be dropping down into the Rift Valley there.
Where did basalt come from?
So it's the East African rifting, the magmatism. And so this is at like 10 degrees north latitude, and we're up at about, the highest point in Ethiopia is in the Simien Mountains area. It's about 14,500 feet. So this picture, we're probably at about 11,000 feet. So it's tropical, but it's very high elevation. And so you have these plants and animals that are found only in this part of Ethiopia. So over here, these are giant lobelias. It's one of these endemic plants. Really cool, so I would be like this tall. And then the charismatic animals, so that is a gelada baboon. So it's a different species than the lowlands baboon that most people are familiar with. These things are herbivores, so they spend their time, they just pull out the grass and eat it. And they're beautiful animals. Unlike the lowland baboons, they're also very calm and they're not going after people. So So really, really unique wildlife. Although Ethiopia, tremendous human history there. Humans have been interacting with the landscape for thousands of years. You know, this is the cradle of humanity. So if you want to go see wildlife, unless you're really interested in these special baboons, you know, you go to like Tanzania, Kenya, South Africa. But the incredible history of humans in Ethiopia. There's the distinct Ethiopian Christianity, so a traditional Ethiopian church, these octagonal buildings. One of Ethiopia's other World Heritage Sites, a place called Lobelia. So between the 7th and the 13th century, these rock-hewn churches were carved out. So this, looking down, oh, I guess I could try to use this. Now I'm all nervous about using this. Okay, so this is the most famous. It's St. George's Church. St. George is a really popular saint in Ethiopia. So there's looking down, and you can see the church just carved out of that, all that basalt. And then here is, you know, looking at it from the side. This is my close collaborator, Bonnie Jacobs, who's a paleontologist at Southern Methodist University. So I did a postdoc with her, started working in Ethiopia, and then we've been working together ever since. Over here, so this is now Dr. Teki Tesfa Michael. He did his PhD with Bonnie, just finished a year ago, and has gone back to Ethiopia and is the only paleobotanist in the country right now. So, you know, it's fantastic. It shouldn't be us white people coming to work on the fossils. We should be working with Ethiopians on their fossils. And then this is a dear friend of mine, Mesfin Mekonnen, and he started off driving for a lot of the paleoanthropology groups, eventually worked himself up to team leader on like the logistical side. So we all go to Mesfin and we say, okay, we want to go into this part of the field for this amount of time. Can you help us get cooks, guards, drivers, cars? So really, really great guy. Okay, yeah, and so, you know, this incredible culture, the unique brand of Christianity. So we do our field work in January. That's the drier season. And lucky enough to be in Ethiopia in January, Ethiopian Christmas is January 6th. And then a bigger holiday than Christmas is the Epiphany, or Timkat as they call it. And so all of the churches have their holy books that regular people don't get to see, except on Epiphany. You have these giant parades where the holy books are taken out and people get to see them. And so here's some of the main most high up religious leaders at one of the churches in Addis. And so they're wearing these things, and the holy books are up there. So they process them out to a big field and then have all sorts of church ceremonies. So just really amazing culture. And then as you get out of the city, Ethiopia has something like, I think it's between 60 and 70 different ethnic groups. And so having spent all this time there, been to different parts of the country, gotten to meet some of these people, gone on tours of various places. So this is down in southern Ethiopia near the Kenyan border. And then here we are in a place called Awash, which is kind of in central Ethiopia, not too far from Addis. And then, of course, adorable, adorable children. Ethiopian children are just beautiful, and I mean, kids everywhere are fantastic. These girls, these are Teki's nieces, so we've spent a lot of time with his family. And then, now, one of the fun things about working is, you know, so we're out in these really rural areas, and you're like doing work, you're digging in the ground, And you're the most exciting thing that's happened in that area in these children's lifetime. So word gets out that, so most African countries have a word for white person. In Ethiopia, it's Ferengi. So the word gets out that the Ferengi are there, and the kids come from miles to see you working. And so all of a sudden you look up and you have this horde of children watching you. So one time I was taking a break from my beloved plant fossils to try to look for vertebrate fossils, and so I'm like digging through the dirt trying to look for teeth, and I'm like gesturing with these kids, like digging and then pointing at my teeth and digging some more. I'm pretty sure the point did not get across. I think they thought I was looking for things to eat instead of looking for teeth, so they were not big help. But they were a big help in teaching me how to count in Amharic. So we spent a lot of time counting. And Amharic is a really, really hard language to learn. So I can count to 10 and that's about it. Oh yeah, and so other kind of unique experiences I had. So this is a buddy of mine who we studied abroad in Tanzania together. And then we lost track of each other for 10 years and then randomly connected in Ethiopia. And so after college, he had gone into the Peace Corps and then was assigned to the Carter Center. And after finishing his Peace Corps duty, he continued working for Carter on Guinea Worm eradication, yeah, which they're so close. So Ethiopia was one of the last 4 countries that still had Guinea worm. It was all the way in the western part of the country. So I went out and visited Adam, and we spent a day doing his job, which is wandering around asking people about, like, do you know anyone who has Guinea worm? If you do you know, like, so it's a year between when you get infected and when the worm starts emerging. I'm sorry, this is lunchtime, this is disgusting. But so Adam goes around and asks people, like, where were you a year ago? And they know. It's incredible, the memory that people can have. So it was a really, really interesting experience. Adam insisted on taking my picture watching the daily guinea worm treatment, and I was not very happy with him about that. So I'm giving Adam the look of death. But they're so close in all of the world to eradicating guinea worm, and also in Ethiopia. So it was,
Where does the guinea worm come from?
So it comes from water. So it has this really interesting life cycle. So it's a parasite, and it infects copepods, which are little microorganisms in water, and humans. And so if you drink water that has the copepods that have the eggs of this thing in it, then you get it. So, the other part of Adam's job, besides like going around and trying to figure out where the cases are and where they came from, is the education of don't drink standing water, passing out these super cheap water filters. It's like a drinking straw with, essentially like cotton in it, and then you suck through that, and the copepods are big enough that you're not going to get those. Yeah, so this sign right here is telling people, don't drink the standing water. That's how you're going to get guinea worm. So there's a lot of really neat experiences, both with Ethiopians and then also with the aid workers. Oh yeah, and this is, again, sorry, lunchtime. So, traveling in Africa, you get a lot of perspective on how well off we are. And so when I visited Adam, and he's staying in there's a UN human rights compounds there, and that's where he lives. And they were building a new bathroom at that time, and so it was like well, that's the bathroom. And so actually I have this picture framed in my office, and whenever I think I'm having a bad day, I look at that picture and I always think, well, at least my bathroom has walls. So, you know, I tell the college students, you know, please go study abroad, because it really broadens your perspective of the world, and you think about what's important and what do we blow up that's not actually important. Okay, so now transitioning into paleontology. And so most paleontologists, they think of Ethiopia and the cradle of humanity. This is where our ancestors evolved. And so I was lucky enough, when I was living over there, Lucy, so 3.4 million year old Australopithecus, so on the line to modern humans. So she had gone on tour of the US and then was coming back to Ethiopia. And quite reasonably, the Ethiopian government said, well, all these Americans got to see her, so Ethiopians should actually get to see her. She's normally locked up in a vault in the museum that no one gets to see. So for a couple weeks while I was there, Lucy was displayed, and it was really amazing to get to see the actual bones. And so so I realized this, you're not going to read all the names, but this is looking at the family tree coming up to modern humans. And now I've circled everything that's known from Ethiopia. So a really, really important place in understanding where we came from. Now, so there are lots and lots of people who are working on this stuff, and it's very competitive and very cutthroat, and that is not me. I very much like going further back in time where you don't have as many people working, and also there's super interesting things going on in Africa then. So like 7 million years ago, that's the earliest hominid ancestor, not known from Ethiopia, but from Chad. But we're going to go back 20 million years to the Miocene, the beginning of the Miocene. And so there's a site in Kenya, it's in Lake Victoria, it's called Rusinga Island, and we have some of our primate ancestors there. And various paleontologists have worked at trying to reconstruct what the environment was on Rusinga. And so, you know, that would give us one data point of what environments in East Africa were available for our lineage to evolve in. So we are looking at the early Miocene in Ethiopia, similar time period to this, and what was Ethiopia like. Okay, so here we have again, so modern Africa. So major features that we see, so first of all, there's the land bridge from Africa across to Asia via the Arabian Peninsula. Another thing you might notice is this very prominent scar across Eastern Africa, the Rift Valley. So these are all relatively recent features. And so Africa, you know, thinking about going really far back in time, and we have Pangea and everything was together, and then you have Gondwana land and Africa and South America were connected, and then once Africa and South America separated, you had Africa as this island continent for tens of millions of years. So from the kind of the mid-Mesozoic all the way until 23 million years ago at the Paleogene-Neogene boundary, there's fossil evidence that you finally have this above ground connection from Asia down to Africa. So we've been working across that boundary. So in the latest Oligocene, we worked at a place called Chilga, and that has this very unique endemic African fauna. So lots of kinds of elephants, lots of hyraxes. There's this extinct group of animals called arsinotheres. So very African. And then you cross into the Neogene, and the mammals are really different, and they're a lot like what you see today. And just emphasizing, so these are plant sites known from Africa, from the Oligocene and the Miocene, and just not very much is known about African vegetation. So everything we do is new and exciting. But most of our modern African animals actually came from Asia once there was the land bridge. So all of the groups of animals that you see up here originated in Asia and came down into Africa about 23 million years ago. And so then we also have that rift valley cutting through modern day Africa. Rifting started in the middle Miocene, so a little bit after when we're studying. Yeah?
So, for some reason I was thinking things would be moving out of Africa into Asia, but what you were saying is they're coming from Asia into Africa and then competing with them?
Yes, and driving a lot of the African things extinct. Okay. Yeah, so it's actually very interesting. So the vertebrate paleontologists are working on this flow of animals, and it's, I don't know the answer. I don't know if anyone knows the answer, but like, why did the Asian things succeed in Africa and the African things not take over Asia? Until humans, of course. Humans came out of Africa and have been very successful. So then we have the rift cutting through Eastern Africa, and today you see very different plants and animals on either side of the rift, especially in terms of things that don't really travel that far. So like amphibians, super different on either side. And then the plants, so you have, it's this maybe the same genus on either side of the rift, but different species. So 15 million years ago, you had this barrier, and it was hard for things to get across, and so evolution takes them in different directions. So our work has been about, so first off, looking across that Oligocene-Miocene boundary when you have the geographic connection and seeing like what's going on with plants then, and then also looking before rifting, what does this ancestral African forest look like? Okay, so that brings us finally to the Mush Valley of Ethiopia. So this is a site, it's about 21.5 million years old. One of the wonderful things about working in East Africa, so it's a very volcanically active part of the world. And so there's lots of ash beds that you can date and know exactly how old it is. So this site is actually 21.73 million years old. Plus or minus 0.03 million years. So this is our colleague from Addis Ababa University, Dr. Mulugeta Faseha, and he is a sedimentologist and stratigrapher. He's really interested in looking at ancient lake deposits around Ethiopia, partially trying to find coal deposits and economically important stuff, but also, as a PhD scientist who's just very interested in the history of Ethiopia, lakes record what the environment was like millions of years ago. So he rediscovered this site. There had been French scientists who mentioned having found fossil wood there in the 1950s. And then Mulugeta went back, and he'd worked with us at Chilga. And so he went back to this place, finds amazing fossils, and tells us, hey, maybe you guys should come study this. So really great group of people working on all sorts of different aspects of the geology and paleontology. So I already mentioned Mulugeta, Neil Tabor from Southern Methodist University, who's a geochemist. So he's working on using chemistry of rocks to tell us about temperature, precipitation, and maybe even paleoelevation. So today this site is at about 10,000 feet, and of course we're very interested in how high was it 21.5 million years ago. Dan was a master's student with Bonnie who worked on the pollen at this site. Michael Engel from Kansas, our insect expert. Several vertebrate paleontologists. This is Matt Clemens. He did his thesis on, master's thesis on some of the amazing fossils I'm going to show you. And then kind of at the bottom, we have here's team paleobotany. So people working on different aspects of the plants. Aaron, very interested in what species things are, trying to tie these 20 million year old plants to modern groups. Rosemary, looking at the molecular signatures of the fossils. Techie worked on paleoclimatology, so he was reconstructing what carbon dioxide levels were like at this place. And then Bonnie and I, kind of the ecologists on the team. And so there's one of our big crews. So we have our white scientists, our Ethiopian scientists, and then the horde of villagers that we hire to help us in camp and then also in the field. And we got our first funding from National Geographic. They really like things like this where you go to someplace exotic and dig up some fossils. And then after getting that seed grant from National Geographic, we wrote a grant to the National Science Foundation and got a nice big pot of money that funded 3 field seasons and then years of work in the museum and in the lab. Okay, so just some fun pictures. So basically we camp in the village cow pasture. So there's Bonnie working away. It's a pretty well-off part of Ethiopia. Average lifespan in this area is about 85 years. So, you know, it's a pretty good place to be. The people are incredibly kind and generous, and I am never worried about my safety there. Ethiopia in general has this incredible culture of hospitality. People just invite you into their home and make coffee for you, and so this is certainly true there. And so there we were invited into the home of some of the people who lived there. So a traditional Ethiopian meal. Well, so here's the injera, so the fermented spongy bread, and then this is gonna be greens. I don't know if we get them here. Like they're thick and tough and kind of like kale in texture, and you cook them for a long time. And then this stuff in here is probably going to be mostly lentil based. Meat is kind of hard to come by in Ethiopia, so you do sometimes have like meat stews, and there's, you know, like one chicken in the stew that gets eaten for a week. So maybe some bits of chicken or goat in there, but mostly a vegetarian based. Okay, so here is looking at the fossil site from above, and so that gully in there, yeah. So you have the modern, so it's called the Mush Valley because the Mush River is cutting through it and exposing these sediments that have fossils in them. So this is what we think is the extent of the fossiliferous sediments. Now, underneath the fossiliferous sediments, you have all of this evidence for explosive volcanism. So this is the very nicely named volcanic bomb. So the big explosion and like big pieces of rock getting thrown into the air by that. So this is lateral to where our main fossil beds are, and you have these big ash deposits. And underneath our fossil beds, so here's a young African man for scale, and you see this tree that's in life position. There's its roots in the tree. So our interpretation, oh wait, sorry. Okay, so this is underneath the fossil beds, and then here's those fossil beds. So they're very finely laminated, mostly black material, very organic, rich shale, and then you have these ash layers bisecting things. So what our interpretation of this is it's a crater lake deposit. So you had this explosive volcanic activity that made a crater that eventually filled with water. And then you have plants growing all around it and animals maybe coming and visiting it. And things dying, falling into the lake, and getting incredibly preserved. So working with Michael, our insect expert, so we found that little bit of insect wing. It's not very impressive, but for Michael, it's good enough to say, ooh, this is a new species of giant winged termites. So we had a paper describing this new species. And then we have some fish fossils. It's not super well preserved, but there it is. And then as far as the animals, these are our poster children. So just these amazing frog fossils. And so this is one reason why we call it a Lagerstätte. So you can see here's the bones of the frog, and then there's the soft tissue. And initially I'd been like, oh man, that frog got smushed. Look at how fat its legs are. But there's wonderful detail preserved, and so we know these are African clawed frogs. If you look at their hands, you can see their little claws. And if you go and Google African clawed frogs, you see they do actually have very fat legs. So interestingly, you get two sizes of frogs. We think it might be males and females. And so there's a bunch of them that are like that big and a bunch of them that are about that big.
Anywhere from 2 to 4 or 5 inches?
Yeah. And then, so not only do we have adult frogs, we have all of these different stages. So here we have our tadpoles, and then you have this half tadpole, half frog. And so you can see, the bones are different colors in different places, and that shows that the bone is in the process of hardening, of ossifying. And then, so these various stages of frogs with tails. So really, really amazing. And then, so not only do you have that morphology preserved, there's also chemistry preserved. So what I'm going to be showing here, so this is a technique of analyzing the chemistry, and you have peaks for different elements. So in this slide right here, so here's where the frog is. This is instead analyzing where the rock is. And so you see there's a lot of silica, aluminum, oxygen. So unsurprising, it's a rock. And not a lot of carbon. And then you go to where the frog was, but not where the bones are. And now the biggest peak is carbon, but also a lot of oxygen, calcium. And then going where the bones are, and just like our bones, calcium, phosphorus, oxygen. So really cool. And even cooler, there are pigments preserved in these frogs. And so we worked with a team who's experts on fossil pigments. And these are the folks who are telling us what color dinosaurs might have been. So they took the pigments from our frogs and compared them with other pigments and did their magic. And they told us that our frogs were dark gray. So it was a little bit sad, but oh well. Okay, and then why I'm there, because there's just incredible plant fossils. So an array of some of the things we found. So leaves, fruits, winged seeds, more winged seeds, floating ferns, legumes, just beautiful, beautiful stuff. And then this is one of my favorite fossils. This is the elephant ear family, a pretty common dentist office plant. And actually, maybe if we wandered around here, we might see things in this same family. But this is Aaron. Here's Aaron's legs and butt for scale. So that's a big block with just these beautiful fossils. And you can see they're attached down there. And we do have the piece where they're attached. So this was just a little thing growing in the mud along the lake.
Are those insect trails? They look like what you were showing.
No, this is just modern mud draped along it.
How do you split a rock that big?
Very carefully. I mean, so we pull the blocks out from the outcrop as big as we can, and then you turn them on their sides, and with a hammer and chisel go all the way around it. It'll preferentially split where the fossil is because that's where there's a crack in the rock. But there's definitely a lot of luck involved. Thankfully, plant fossils are very abundant.
Modern-day roots there.
Yeah, and they'll exploit those cracks as well. Okay, so we've been able to put names on things, unlike what I talked about last night. So we have this beautiful fossil that's in the yam family. And then some of Aaron's work. So we have this particular winged seed. It's in the legume family. He's assigned it to a modern genus and then plotted, here's where you find this genus today. And then there's where we found it 21 million years ago. So as you start doing more and more of this, you can tell really nice stories about how the ranges of organisms changed in Africa over the last 21 million years. Okay, well we had last night, so not every fossil can be identified, so we will just describe the different types of leaves. And we rely very heavily on the shape and the veins, but it can be really hard with tropical leaves. Tropical leaves almost all have smooth margins. They're elliptically shaped. They have what we call drip tips on the top. Veins can be hard to distinguish among the tropical taxa. But here, so we have the cell structure preserved. So the waxy coating, the cuticle on the surface of the leaves is preserved. And so the cuticle has the impression of those epidermal cells. So we have the upper surface of the leaf, and you can see these are bases of hairs. And then the lower surface of the leaf, and you can see some of those hairs. And then you can also see the stomata, which are the little noses for the leaves. So they're breathing in CO2 and out oxygen and water vapor through those stomata. So there's one nice example. Here's another one, this lobed leaf. And here, oh, I've got it backwards, but here's the upper surface, and it's got these weird hair structures on them. And then the lower surface with all the stomata. And so this was really important in helping us differentiate how many different things we had. And there's tons of legumes there. So here's our what we now know are our 3 most abundant legumes. And I mean, they look kind of similar, and there's a lot of variability in what shape they are. So here kind of you're seeing end members of what shapes they are. But it was a real puzzle trying to differentiate them from first principles. But once you have the cuticles, you see that cell structure is so different, and you would never mix up those pictures up at the top. So this has been really helpful. The way these images are taken, this is false color. So it's a technique, it's called epifluorescence. And so you have a microscope and you shine really intense light of a certain wavelength that'll excite the organic matter. And so for plants, it's blue light. So you shine really intense blue light at the fossil. So the organic matter is excited, and then it gets excited, and then it emits the energy again, and it'll emit in the green or yellow spectrum. So that's why we have those colors of image. And then parts where you have more organic matter are going to shine more brightly than less, and so like seeing where those cell walls are. So really, really, really cool fossils. And then another thing we've been working on, so using a combination of cellular morphology And so see, like these cells, they have very sinuous cell walls, very undulatory. And some modern work has shown that plants that grow in full sun tend to have straighter cell walls, and ones that grow in the shade tend to have more undulatory cell walls. And so there's a way you can quantify how much undulation you have. And that's what we would do on this axis here. And then also the chemistry of the leaves differs based on where they're growing. If they're up at the top of the tree in the sun, the carbon isotope values tend to be higher, versus in the shade they tend to be lower. So we've been working on doing this combination of cell morphology and chemistry and trying to figure out exactly where in the forest these different leaves are growing and the different species are growing. Okay, and then, so for those of you who were there last night, so this is the site where we found the scale insects pressed onto the leaves. And so just a close-up. So, you know, these guys are, some of them you see with the naked eye, they're maybe half a centimeter big. And then other ones are quite small and you would miss them. But then when you shine the epifluorescence at them, then they glow very nicely. So at least 4 different types of scale insects on these leaves.
Never found any in Wyoming?
Nope. Well, at Big Cedar Ridge, that 71 million year old forest that was buried by ash, there is a scale insect known from one of those leaves. It was really funny. So some scale insects look a lot like trilobites. So the marine critters that lived in the Paleozoic. And my colleague who found that, he's like scanning along the leaf, and he's a paleobotanist, so he doesn't think about arthropods very much. He's like, wait a minute, there's a trilobite on this leaf. What's going on? And then he was like, wait, maybe it's an insect, not a trilobite. And then he got the insect guy, and it was one of the scale insects. Okay, and then, so we sometimes have the insects preserved. We can also look at the feeding traces on these leaves. So I'll just get them all up. So all sorts of interesting feeding traces. So things that chew along the leaf. These up here, we're looking at various gall types. This right here, it's something we didn't talk about last night. It's their oviposition scars. So insects laying the eggs in the leaf. So those scars being made by the ovipositor, the bud of the insects depositing the eggs inside the leaf. Okay, so one of the big science things that we wanted to do was, besides documenting, okay, so what was this ecosystem like during this really interesting time period before the rift, we were interested in how much did things change through our sequence of beds. So capture small scale changes, and then when you're looking across like millions of years and seeing how much change is there, that can help you to figure out, like, is this just regular small scale change, or did something big happen? So we looked at these, we picked 6 stratigraphic levels, and what we did was we would just completely excavate a 5 centimeter layer and then bring it back to the museum. It's really hard to ID these fossils in the field. Also, you might have noticed in the image of the fossil site, you know, the river's cutting right through. It's really wet rock. And you take really wet rock and you expose it to air at 10 degrees latitude and 10,000 feet, and the rock just cracks immediately. So we didn't have time to study it in the field, and so we're like, well, we're just gonna excavate the layer, wrap it up in toilet paper, let it dry slowly in the museum, and then come back and work on it slowly. So that's what we did. So 6 stratigraphic levels, carefully ID'd in the museum using epifluorescence, over 2,200 leaves from these 6 different layers. Identifying how many different leaf types we have, and then looking at the feeding. Oh, and one more thing to mention. So you guys saw in the picture mostly black rocks, but some little white layers mixed in. So, little volcanic events. And then, so this right here, it's about a 10 centimeter thick ash layer. So perhaps a more significant volcanic event. Although probably still, pretty far away from where we are. So we thought it would be good to, see what fossils were preserved in that layer itself, and then see how things are different before and afterwards. Okay, so I really like this plot. But so what I've done here, these are the 49 different types of leaves you see, and then it's just showing how abundant they are at our 6 different levels. So you'll see at all of the levels, this thing that we call legume 1 is the most common, but it's much less important at the lowest level than it is at these higher levels. So this is just kind of a nice way to see how composition changes. These J numbers, that's a way to quantify how even the distribution of plants was. So if you had exactly the same number of every leaf type, your J would be 1. So in general, you see these are all pretty dominated by this one species right here. But this one, it's much less so than at the others. Okay. So we saw an ordination last night. What's going on here is plotting the different sites by their composition. Okay, one thing I didn't mention, we didn't just dig one hole and collect 5 centimeters from each strat level. We did it in 3 different places, so we could capture a little bit of the spatial heterogeneity. So this plot right here is showing those individual holes, and then I've circled showing them being from the same stratigraphic level. And then here's kind of distilled down just by strat level. And you see, the floras are really pretty similar as we go through. In fact, levels B, E, and F are so similar. So here's those individual holes all plotted on top of each other. And if we lump the data, they all plot on top of each other. So not a lot of change through this section.
Okay.
Yeah.
5 centimeters is really little.
Well, it's like that thick.
2 inches.
2 inches.
So, but laterally, how much of that did you take?
So our individual holes tended to be maybe like that big, and then we would do 3 holes that were something like 25 feet apart.
So like 3 by 2?
Yeah.
Could you repeat that for that? Because they can't see you.
Oh, do I need to be on here too? Or
at least say it.
Yeah, so each of these here, so it's a 5 centimeter thick layer, or like 2 inches, and then that was collected from like a 3 foot by 2 foot area. And then those areas that are the same stratigraphic level are maybe like 25 feet apart. Okay, so this is the most complicated slide, the only big data slide. So lots of things going on in here. Let's first start by looking at this one right here. So this is looking at how the number of plants that you find changes through sites. And the number of plants that you find is going to be strongly impacted by the number of leaves that you look at. So these things here, these are called rarefaction curves. And so based on a given sample size, how many different plants would you find? So you can like pick a sample size here, draw a line up, and we would see that that highest stratigraphic level, level F, is way lower than all the other ones. Our level C, which is the one that was in that thicker volcanic bed, so we weren't able to get very many fossils from it, but it's that line right there. It also seems to be less diverse than the other ones. Okay, so this is the same thing, but now instead of looking at the number of plant species, we're looking at the number of damage types. So a proxy for the number of insect species. And you can kind of compare and where do things fall out. And it's kind of interesting. So that site that had not very many plant species actually has a pretty high diversity of insects. And then, so this one right here is that lowest stratigraphic level, and it has low diversity of insects. Okay, and what's the one? Okay, so let's look at this one right here. So in work that I've done, this is something that I've found to be important. So specialized damage frequency. And we can look at the types of feeding marks on the leaves, and based on what types of damage, you can get an idea of what kinds of insects are eating things. And by that I mean, are they insects that are generalized feeders where they can just eat everything? Or might they be insect groups that are really focused on one particular plant species? So things like gallers and leaf miners, they always target the same plant species. And in later successional forests, you tend to see more specialized feeders versus earlier successional forests, more things that are just capable of eating everything. And so we look here, and so that lowest level A has pretty low specialized damage frequency. So kind of putting this all together and thinking about what different levels of diversity mean and what different levels of damage mean. And so what we think, so we're going through, and you know, we've got our one date here, and you know, that's what we've got. And chances are, like, if we were to date up here, we wouldn't be able to do it well enough to get within that 0.03. So we don't know exactly how much time it is. I would guess we're looking at something between 10,000 and 50,000 years. And so here, level A, so it's this forest recovering after the big volcanic explosion that created that lake. And so things were probably, so this is the explosion that we think created the lake, very turbulent landscape after that, and then the start of regular lacustrine beds. So we think it's a pretty early successional forest. And then you come into level B, diversity increases, you have more of this legume 1 coming in, so later successional stages. And then at level C, you have this small perturbation to the environment. So again, very low diversity, but lots of different things spread out across the landscape. And these two things that we've identified from it are things that tend to be in modern African early successional forests. And then, so recovering from that, and it's very similar to what it was here. And this is our level that has the highest diversity. And then things just sort of continue, but legume 1 just is really taking over the forest at that stage. So that's our story about, you know, this change through time. So we capture, you know, what change do you expect in kind of a normal, say, 50,000 year interval, and then that's really important in us interpreting those bigger changes through more time and looking at different sites. And so just to close, so we worked with Karen Carr, who's an amazing paleo illustrator. And so here's a reconstruction of that mush forest around the lake. So the frogs with their chubby little legs. Here is this winged seed Newtonia, so it's in these big pods, and then the seeds are hanging out. And the very lush vegetation down here, those are some of our elephant ears. And then, of course, because I'm part of the team, so Karen puts little bug bites on those leaves. So that's Ethiopia. and the pre-hominid fossils of Ethiopia. Did you have a question?
Yeah, I mean, you've got the frogs. When did other animals start appearing in that whole thing?
Yeah,
Because did you have fossils that showed anything, or?
Yeah, well, so a lot of the beds here are not good for preserving animals. And it's pretty amazing getting all those frogs. There obviously must have been a lot of frogs. I was hoping to one day split a rock open and find a bat there, because we have bats in the Green River Formation. There's a Crater Lake deposit in Tanzania called Mahenge that's from the Eocene, and it has the earliest bat is out of Mahenge. Or wait, no, the earliest African bat is Mahenge, but Green River is older than Mahenge is. So I was really hoping to find a bat, but I never did. So you can see, like on our strat chart, so there are levels that bones were found. So in this level right here, there's frogs, there's fish. Up actually above this, we do find some mammal bones. We've got a tusk of maybe a warthog-like animal. There's a few croc teeth and crocodile teeth and vertebrae that have been found through here. But I think we're in the wrong environment to preserve bones well. So unfortunately, with the exception of the frogs, we don't know a lot about what animals were around here.
Have you compared the leaves from this Mush Valley to say, Cameroon or something, or other parts of the world that may have been in similar circumstances at the same time?
Yeah, well, I mean, that is definitely something we would love to do. And you saw the map of Africa and how few fossil sites there are. So, we've compared our things with the older sites in Ethiopia that we've worked on, and there's very little overlap. Now, some of that might be because we're in different environments. Some of it might also be the time driving it. There's some newly reported plant fossils from West Africa, from, where are they? I think they're working in Cameroon. And so I'm really interested to touch base with those folks and see how ours compare. Yeah, because getting at this, today we know things are so different. And we are, quite close to where the Rift Valley is today, and we find things that are closely related to things. So we're on the western side of the Rift, and we do find things that like the closest relative to some of these is on the eastern side of the Rift. But yeah, it would be great to have more spatial coverage on the African continents.
Could you give a quick tour of the key to that, basically?
Oh.
What some of those
Yeah, sure.
the pattern symbols mean?
Yeah, so first off, looking at, how far out things go, and so this is based on the grain size of the rock. So,
How resistant?
no, it's not resistant, it's just the grain size.
Grain size, okay.
Yeah, so all the way out here are conglomerates and then sands, and then now our sedimentologist who did this distinguished muds, clays, and purely organic layers. So even though this does come out to here, if you follow it down, it's in the mud category. Then the color is a representation of what color the rocks actually are in the field. So you do have some slight color differences as you go through the beds. And then these white things that stick out, those are the ash layers. And some of those ashes have a very nice crystalline structure and were probably ashfall. But then this one up here, it has these little symbols in it that tell us it was water reworked. Those are trough crossbeds. So little sedimentary structures telling us that the water reworked the ash. So like a little delta coming in, depositing that. And then leaves are very abundant throughout, a few bones. Not entirely, I don't remember what those boxes are. Oh yeah, so in this like very turbulent time, so trough cross bedding, these are the symbol for symmetrical ripples, so where you have back and forth motion of the water. Yeah, so significant in this interval, significant water reworking of the sediments.
When was the age of the beginning of the Rift Valley?
It's mid-Miocene, something like, I think about around 15 million is the inception of rifting, and so it starts in the north and then gets pulled south.
And that's pulling two continents apart.
Right, yeah. Yeah, so if we were to...
Somalia and the African.
Yeah, where's...
So 15 million years, I would have thought longer.
Yeah. Oop. So we pull up Africa again, and then so there you can see there's the rift, and so the
whole
Somali
part, yeah,
being pulled off.
Yep.
And then someone mentioned the triple junction earlier, so
Yeah.
you know Arabia is going up that way, and yeah.
What makes it hard for plants and animals to get across the rift? You said it divided the forest into two.
Yeah, so in the rift, so it's a very arid environment, and today, you know, it's all grasses in the rift. It's too dry for like the big, you know, on either side of the rift, that's the rainforest belt, and those trees, you know, this environment in the middle, it's inhospitable to rainforest trees.
What makes it dry?
Yeah, well, it's definitely, okay, so it's lower in elevation. Also, I mean, so we haven't really talked about like how climate's been changing in Africa, and if we continued over here, India would be over here, collided with Tibet, and having a huge impact on climate all across this region. And so you have more of a monsoonal climate in Eastern Africa. So a pronounced rainy season. So there's the long rains, and then there's the little short rains. So I think it's this fortuitous combination of the drop in elevation here, but then also climate having changed as that occurred.
Would you expect Africa and South America to have similar flora at those particular times? Because you said it split apart.
It would certainly be more similar than comparing Africa with Europe. Yeah.
It's already been apart for quite a long time now.
Pardon?
It's already been spread apart for quite
Yeah. So, but yeah, it's been 50 million years of separation.
They showed that they were,
Oh, it does.
you know.
Yep.
That was Wegener's,
Oh, no.
Wegener's big thing.
Um,
The same.
But actually, so there's like, so looking at the modern tropics, so like you have tropical rainforests in South America in the Amazon, and then like across the Guinea, Congo area. So this whole region in Africa is the jungle. And then in the Southeast Asia, you have the tropics. And so like one of the things that's super interesting, so Africa is often considered the odd man out compared to South America and Southeast Asia. Africa has lower diversity in the tropics today. And then also some of the plants that are really important in the New World tropics and Southeast Asia, like the palms, are not very diverse or important in Africa. So also, like, part of what we're trying to do, and I didn't bring that up earlier, is look at the history of African vegetation. And so if we were to go back to the Cretaceous, and people have done a lot of pollen work in all three of these places, a lot of it driven by the oil industry. So beautiful Cretaceous pollen records from these three continents. And the diversity in these forests seem pretty comparable. Palms were really important in Africa in the Cretaceous. But then something happened between the Cretaceous and the present day to make Africa really different from the rest of the tropics.
It's sort of a cultural thing. How you don't speak the local languages other than counting 1 to 10, I guess.
Pretty much, and say hello and thank you. And
so I mean, is it very common to find people away from the cities that speak English?
Well, so no, it is not. And actually, you know, I'd mentioned how there's something like 60 different ethnic groups and they each have their own language. And so even like the people from Addis who come out in the field with us, they might not be able to communicate with the locals because they speak a different language. So Ethiopia requires all foreign scientists to bring a government representative in the field with you, and you actually take one, and so these people, they're the, what's it called? The antiquities departments. There's a longer name for it. So they're mostly trained as historians. So we've had some really bored historians out in the field with us. I mean, they're lovely people, but they're like, oh my God, what are you doing? And then they go off and talk to the local people and then, you know, tell us about like what life is like there and what's the village gossip and things like that. But so anyway, so we're required to take someone from the antiquities department in Addis out with us. And then recently they have also said, no, also you need to bring a local person. And this is actually really good. That is the really useful person because, and they're almost all men. Well, they're all men. I've never worked with a woman. And so he'll know English, Amharic, and then also whatever the local language is. And then this is a member of that community. And so the locals know this person, respect this person, and then, you know, he can also tell us like, okay, here's what is reasonable to pay someone to do these things. And then we know we're paying a just wage.
That person probably would be useful if these, I'm going to call them tribes for lack of another label, but
yeah.
Do they have oral history? And does
yeah.
then give you that so that makes
when they're bored and they go and talk to them, yes, we do get that, or some of the oral history. Yeah.
As a female scientist, what are the challenges that you see compared to your male compatriots?
Definitely a lot harder to be taken seriously. You know, people always assume that I'm the student and whatever white man is with us is the professor and I'm his student. I'm not allowed to carry things, which I guess is a blessing and a curse, but like I have to fight to be able to actually carry my hammer with me. I have a lot of people telling me how to do my job.
Your fellow scientists or the Ethiopians?
Both.
Why can't you carry your own tools?
Because women don't carry tools.
except on their heads.
But they carry
Except on their, well, yeah, but see, there's this, you know, it has to do with the people you're working with as well. So, yeah, the working class, or not working class isn't the right word, but the very low income people, yes, it's the women who are doing all the heavy lifting, and we saw it in that picture. And I've seen, so Addis Ababa is, it's a city like in the middle of the basin, and there's the mountains around it, and the mountains are forested. And you see women walking down from the mountains with like tons and tons of logs on their back. But then you move up into like the middle and upper class Ethiopians, and so those are the people that we're working with. That's who the government workers are. And so then the standards switch, and science is what men do, and digging up rocks is what men do. So it might be different if we were, actually working with the very poor people.
Is it like that in the college there too, that the students, are there female students in the sciences in the college?
There are a few now. I have not met a female science faculty member. I've met, a bunch of the geologists, some of the biologists, always men. In the museum, all of all of the curators are men, most of the junior curators are men, but there are a few women. So I do try to, when I'm over there at the museum, reach out to the women coming up through the ranks. Ethiopia is a very hierarchical society, and the professors are treated with just such tremendous respect, and it's something, there's a barrier to like questioning your professors, and this has been hard with some of the Ethiopian students that we've brought to the US and had in our classes and our grad programs. They come out of a culture where the elders know best, and what the professor says has to be right, and then you're trying to teach these students. And the same thing happens in America too, but getting people to question what they read, and just because you read it in a paper doesn't mean that it's right. Just because I said it doesn't mean it's right. We do science to try to prove things wrong. So there's definitely been some cultural challenges.
That isn't just in Africa. You see.
No, it's not. Yeah, I see it. It's stronger in Africa than it is here. But no, it's certainly. I find like the biggest struggle with American students coming out of college into grad school, teaching them to think critically, to not just believe something because it was written in a paper.
So there must be a lot of corruption in Ethiopia, like the rest of the continent. Did you run into that yourself?
No, so in college I worked in Tanzania, but that doesn't count because you have no idea what's going on when you're in college. So as a professional, Ethiopia is the only place that I've worked. But Bonnie has done a lot of work all across East Africa, and Ethiopia is the only place where she's never paid a bribe. So, there are rules and you follow the rules, and there is no culture of bribing people. There is understandably, the culture of like, if you want to come in and study our stuff, you need to contribute to the economy. So, but it's very above board. Like, you hire the antiquities rep from Addis, you hire the antiquities rep from the region you're working in. You pay the museum a fee to work there and to have bench space. So I've personally not experienced corruption.
And the fossils you collect there, do they stay in
Yeah.
Ethiopia, or can you bring them back?
Nope. Well, so with hours of begging and pleading, and I will admit crying in various people's offices, there is a procedure to borrow fossils for a short period of time to study and then bring back again. You have to make a really strong case of like, here's this equipment I have in the US that we don't have here, and so I have to take it. We're really the first paleobotanists who have been working in Ethiopia, and so it's been a struggle to get them to understand, like, I'm not asking to borrow Lucy. These are not priceless artifacts. Like, if the plane crashed and we all died and the fossils got destroyed, you could go back to Mush and you could collect probably the same species. And, so we've spent many years now having these discussions that, yes, these fossils should absolutely stay in Ethiopia. They're your country's history. But if we borrowed them, and something happens, it wouldn't be the end of the world. So Ethiopia is very proud of the fact that it's the only country that wasn't colonized. It was briefly conquered by the Italians in the 30s. Haile Selassie, the most famous Ethiopian, went into exile and then came back to Ethiopia again. So there was this brief Italian occupation, and the Italians just stole all sorts of archaeological remains. And so that is very much on the mind of the Ethiopian government, and they want to make sure that their history stays in the country.
What's the current elevation of that Mush Valley site?
About 10,000 feet.
And what did you or your patriots decide the depositional elevation was?
Still in progress. Paleo elevation is one of the holy grails of geology. There are lots of different techniques that people have proposed to try to get at it, but the techniques all have a lot of assumptions and very big error bars. So
what's the guess though?
Well, it's certainly, it wasn't sea level. Probably wasn't 10,000 feet. Maybe kind of midway in between. How's that for a hedging answer?
Plus or minus 5,000 feet.
Yeah. Well, actually, so I just Bonnie just texted me a picture of a talk that Neil Tabor, our geochemist, is going to give on Friday, and the name of the talk is like scratching my head over the height of the Ethiopian plateau. So. She's in the same department as Neil, so she'll obviously go to the talk on Friday, and we'll see what Neil's latest thoughts are on the height of this.
That's quite a range. Yeah. What else? Anything else? You're going to get more detail from Mr. Surdom over here about what to do in the Washakie Basin?
Well, we're going to show her some next summer.
Yeah.
Places where really leaf rich, and they're top sets on deltas, upper Miocene deltas, and they're just leaf trash everywhere you look.
Neat.
But I would say